हाय स्टूडेंट्स। इस वीडियो में हम समझेंगे कि ब्लड ग्रुप क्या है। यूजुअली जब भी हम ब्लड ग्रुप पढ़ते हैं तो हमें ये कंफ्यूजन रह जाती है कि एंटीजेंस कहाँ हैं और एंटीबॉडीज किस ब्लड ग्रुप में है। इस कंफ्यूजन को हटाने के लिए लेट अस अंडरस्टैंड ब्लड, एंटीजेंस, एंटीबॉडीज एंड व्हाट आर देयर फंक्शंस एंड एग्जैक्टली वेयर दे आर प्रेजेंट। तो हम इसे आसान करते हैं। लेट अस स्टार्ट फ्रॉम द ब्लड। So first of all, what is blood? Blood is a liquid connective tissue. Blood is a liquid connective tissue. It is also called as softest connective tissue which is present in human body. Blood consists of two parts. One is the particulate part. Particulate means particles का बना हुआ है. Particulate part. The other one is fluid part. Now, fluid part is basically plasma, which is about 55 to 65% of the blood, and particulate part contains, that RBCs, which is erythrocyte, WBCs, these are leukocytes, and platelets, these are thrombocytes. Our concern is RBCs. So, red blood cells. Now red blood cells, they are biconcave in shape, biconcave disc-like structures. They are biconcave. Why they are biconcave? Because of the biconcave nature, from both the sides they are pressed and easily float into the plasma. Correct? Now RBCs, which are biconcave in nature, let's make a two-dimensional structure, because on the board three-dimensional structure cannot be made. It is containing hemoglobin pigment in it. These hemoglobin pigments, a single RBC contains about 300 million molecules of hemoglobin, which can capture about 1.2 billion molecules of oxygen. Remember that. So, this is the RBC. On the surface of the RBCs, antigens are present. On the surface of RBCs, antigens are present. Let's make a diagram to understand what kind of antigens are there. Suppose there is a RBC which is having an antigen. Its shape is like a triangle. You can say, when we write A, the same kind of triangle comes, although a horizontal line comes in between the A. But we will neglect this horizontal line, and this triangle shape is basically an antigen which is present on the surface of the RBCs. So if it is resembling with the structure of the A, as you can see, this is also a triangle and A itself is a triangle. So it is a antigen A. It is a antigen A. Correct? Now suppose we have another RBC which is present in another blood. Just to make it more simplified. And this is the hemoglobin pigment which is present in it. I'm making a rough diagram because diagram is not important. And it is also having an antigen present over it. How we write B? We write B like this. B like this. Right? On both the sides, they are circular in shape. So antigen, one is having A-shaped triangle and other is having a dome shape of one of the bulge of B. So if there is triangle, it is antigen. If there is one bulge of B-like structure, it is antigen B. So if we take a blood and on the slide glass, if we place it and make it more diluted and then see from the microscope, we can see the structures available on the RBCs. If it is triangular shaped structure, it is antigen A. If it is a dome shaped structure, it is antigen B. This decide whether the blood group is A. If the antigen A is present, the blood group is going to be A. If this dome shaped structure of antigen is present, the blood group is B. Now suppose we have another RBC. Suppose we have another RBC. We cannot make three-dimensional structure on the board, so I am making a two-dimensional structure by which you can at least have a biconcave image in front of you. Now another RBC is there in another blood which is having both the antigens present. Both the antigens present over its surface. Now it is having antigen A as well as antigen B. So means both can be present. This is antigen A, this is antigen B. Now, three cases we can see. One having antigen A, having triangular shape antigen. One RBC having antigen B, the dome of the B which is present over here. One RBC which is having antigen A and antigen B, both are present. And another RBC, so how many pigments of hemoglobin are present? 300 million. Remember that. Another RBC which is having no antigen present over it. There are 4 RBCs, so 4 kind of blood groups are there. The blood group which is having antigen A is blood group A. We can find by triangular antigen present on the surface of RBC. Another blood group having RBCs antigen of dome of B shape. So one of the dome of the B shape, it is blood group B, and this one which is having both the antigens A as well as B, we will say this is AB blood group. And the RBCs which is not having any antigen is blood group O. So there are 4 blood groups: A, B, AB and O. A is having antigen A, B is having antigen B, AB is having both the antigens A and B, and O is having no antigen. I hope you understand how we decide the blood group. In the next video, we'll be discussing about the antibodies which are present and why they are present in a particular blood group. Thank you.